Chief Executive Officer, SciGenom Labs Private Limited. Let's welcome Mr. Sam Santhosh. And we have three speakers. Let's welcome Dr. A.B. Oommen, Director, International Business Expansion, Neogen Corporation. Let's also welcome Dr. Ramesh Hariharan, Founder and CTO, Strand Life Sciences. And we have Dr. S. Vidya Sagar, Assistant Professor, Department of Radiation Oncology, University of Florida. Now, may I briefly introduce our moderator, Mr. Sam Santhosh. Ladies and gentlemen, Mr. Sam Santhosh is the founder and CEO of SciGenom Incorporation, a genomics R&D organization that conducts research work aimed at understanding the role of DNA sequence variation in human health and disease. Sam Santhosh was the CEO and Managing Director of California Software Limited, CalSoft. He founded CalSoft in 1992, and under Sam's vision, CalSoft rapidly grew from a niche player in the Silicon Valley to a global player with over 1,200 employees and presence in 8 countries. Sam is passionate about science and technology and believes that harnessing the power of science is critical to innovation and wealth creation. He believes that science will directly drive economic development as well as social and environmental well-being in the 21st century. It is a strong belief that has led him to make the move from software sciences, where he spent the last 20 years of his career, to a future that blends science and technology with business. Now, may I request Mr. Sam Santhosh to give his opening remarks. Thank you.
Welcome. Thank you. You must be pretty surprised, to see a session on science in an entrepreneur conference. So I felt that I owe a brief explanation to why we have a session like this. 2 parts, basically. One, why science and business? So that's a pet theme that I can actually talk for many hours, but I'll just bring it down to a few sentences. If you see the industry in the last maybe 20th century, you can see that the big thing that happened across all industries was the influence of technology. So the whole business world was driven by developments in technology. So it's a technology driving business. Science was always there, but science was seen as one initial start to give the R&D departments of companies or governments, which will do science and will come up with inventions or discoveries, which will then take 20, 30, 40 years to convert to a technology which can be used by you and me. And if you look at the steam engine, the automobile, electricity, the telephones and all that. So that is the way business was done, and the business would make money, and some of the money would then go back to the research and development, which is a funding for new discoveries. So that is the model, traditional model of progress in the 20th century. But now as we get to the 21st century, that model is changing. So the pace of progress, the pace of science is so fast, so you just can't wait for these 20, 30 years for a scientific development to get converted to a business opportunity. So we will see, as these examples you'll see, and you'll see more as you read and as you look into the industry in the 21st century, we will see a lot of scientific developments being converted quickly into business opportunities by talented entrepreneurs who will not wait, and who will, whether we like it or not, these will be opportunities that people will take advantage of and bring and convert them into business. And that is driven by many reasons, not just for the wealth creation, but that is very critical for solving a lot of the challenges that we face as a species. So, the last 20th century, we did so many things without really understanding the damage that we were doing. The science and the business were so far apart that the scientific developments were taken advantage of and converted into many things that we all needed. The clothes we wear, the cars we drive, the food we eat, all was done without really fully understanding what damage this was also creating to the environment, to our earth, to our the air that we breathe. And also we are too late to realize that they just couldn't solve all of our problems. So the 7 billion people that we have now, and which will grow to maybe 9 billion in the next 50 years, we just can't have the last century's approach to our well-being. We will need to come up with a completely different type of answers, and what that will be driving will be science. So here we have three entrepreneurs. So I won't introduce them in detail because we'll have a brief introduction from the lady as each one of them comes to speak. So, but I just want to highlight some things. Why invited the three scientists here. They're all from Kerala. So they're all, I think, mostly from Trivandrum and Cochin. Did their initial education in India, then went for their higher studies, to the US. From very well-known institutions, took multiple degrees. You'll find a doctor with a couple of PhDs. You find a IIT product. And they went out, and not only did they excel at their fields of endeavor, which they studied in science, but they were able to take up ideas from that and build businesses. And many of them, not one, maybe some 2, 3 businesses they could build and successfully build it and take it down to a situation where they're able to sell it and create wealth. So this I felt, and Ty felt here, that this is such a great opportunity for us to not only hear the story, but be able to see that how all of us can try to emulate and learn from them, and try to create other good stories like this on our own. So welcome to the session. We might be forced to keep you a little late for your lunch, but the lunch is for 1 hour. And we'll make sure that food is left for you, so nobody will go hungry. So I first welcome Dr. Abe Oommen.
Ladies and gentlemen, Dr. Abe Oommen is the Director, International Business Expansion, Neogen Corporation. Abe Oommen is currently Director of International Business Expansion with Neogen Corporation of Lansing, Michigan, USA. Neogen Corporation develops and markets products dedicated to food and animal safety. Prior to this, Abe was General Manager of GeneSig, a division of Neogen Corporation. Abe was the President and the CEO of GeneSig until its acquisition in 2010 by Neogen. From its humble beginning in 1998, GeneSig has grown to be the largest genomic services provider in the world for the agriculture and companion animal market. Abe has a PhD in molecular genetics biochemistry from the University of Kansas. He also did postdoctoral work at the Nobel Foundation. Now may I request Dr. Abe Oommen for his presentation.
Start? Okay, can you hear me now? Okay, great. Thank you for the introduction. Thank you, Sam, for the introduction and also inviting me. What I'll do is I'll give you a quick introduction to what Neogen is, which is the parent company of the company that I founded, and then tell you a little bit about what GeneSig does. And then I really want to talk about entrepreneurship and what I have learned as an entrepreneur, and some maybe bits of wisdom, I don't know, but it depends on how you see and hear what I have to say, but mostly from my experience. Neogen is actually a food and animal safety company. It's a publicly traded company under the NASDAQ. It's about 200 million in revenue. We have 750 employees spread over 110 countries that we sell our products to. Most of what we do is diagnostic test kits for food safety, in the sense that it's kits that look at allergens and, I think I'm gone, contaminants in food. We also have a slew of products. Okay, it still seems to be working. For animal safety, everything from intervention products, in the sense that from syringes and needles all the way to genomics. And how does it all connect together? Basically, we do everything from the farm to the fork. Somewhere your food is touched by something Neogen does. At least that's our goal, to have that touch with every step of the way. So all of this goes to food security. Like Sam was alluding to, the population of the world is going to be about 7 billion people, and the way we do agriculture, the way we do processing of food, it all has to change. So what the company does is go into the areas that matters in that business, in food security, in the sense that we work with food safety, animal safety, and then production efficiency is where genomics comes in. And that's how they acquired my company, okay? And it sort of makes sense as we go. And we have facilities around the globe. Oh, okay, that's where it is. I was wondering where the slides are showing up. We are in China, in Europe, and Mexico, and Brazil. Right now we have direct presence in those countries, but we sell through about 120 countries, actually. So what are the type of products? From the farm gate, we have intervention products, like I said, veterinary instruments, we have vaccines, and we also sell a whole bunch of rodenticides and disinfecting chemicals that are used for cleaners and disinfectants. We also have diagnostic products, everything from toxins to allergens and drug residues. Toxins, for example, shellfish toxins. Some of you might be allergic to that, so we can test for the presence of it, and also we can test for if you're allergic to it, if you're making antibodies to it. Drug residues are a huge problem in China, and I know from talking to people in India as well, where people add antibiotics to milk. We have kits that will allow you to test for those. Sanitation is a big thing with food safety right now in India. So if you clean a bench that you're working on with food, you can clean it and say, well, is it clean? You can just swipe something and put it into a little device that we have, which you can see that little blue thing on the middle there. I don't have a pointer to point to it. And it'll tell you whether the surface is really clean or not. It's actually looking at ATP. Okay, so basically all of this can be integrated for enhanced food production through genomics. And that's where GeneSeek comes in, the company that I started. So I'll give you a little bit of introduction to that company. This is about the time we sold the company. It's about 32 people in 2010. And that's the picture I have. I don't have a most recent picture of it. But you can see me standing at the very right end of it with more hair than I have today. But we are today, I can definitely tell you, we are the market leader in genomic testing for the ag and companion animal market. We started with one student intern, myself and a professor from the university, Daniel Pomp, with one part-time student intern who worked in the evenings, and I would run from my job, day job, in the evening and work till 1 o'clock, 2 o'clock in the morning, just continue the cycle all over again. And then got tired of it very quickly, and then quit my job and went full-time. Very risky thing, but sometimes you have to take risk. And we'll talk about that in a few minutes as well. Today, we are about 100 employees, 8 of them with PhDs, and 2 of them are doctors of veterinary medicine. We started with 500 square feet, and when Daniel and I looked at that space, we talked to each other, how are we going to fill this massive space, 500 square feet? We thought it was huge. Today we are in a 15,000 square feet lab. We have just purchased a building which is 40,000 square feet, and we're just getting ready to move into it by hopefully March. And we also do 1 million samples, and you may not think that's a lot, but imagine if you are able to get that many samples, that's about 25,000 samples a week. You have to get DNA out of it and get data and send it out so that the quality is also very good. It's a lot of work. It's a lot of physical manual labor, actually. Okay. So what do? We do these things that I've listed there, high content SNP profiling. You will know that here very quickly. Marker-assisted selection, where you can use markers, DNA markers, to select the right kind of animals. For example, if you look at that mice in the bottom there, there's fat mice and skinny mice. You can actually, that's a good example I thought you would understand real quick. You can run DNA markers to select the thin mice versus the fat mice. We work with all those species that I've shown there and more. It gives you an idea of the type of samples we work with, everything from plants and fish. Fish actually is a fairly new thing in the last 2-3 years. We have been doing a lot of work with salmon and shrimp industry in Latin America, but salmon primarily in Europe and Latin America. Okay, so in 2010, Neogen acquired us. And what I started as GeneSeek got morphed into this logo, and the goal of the company changed as well. And this is all the things we do, everything from project management, and getting your samples in, running them on certain platforms. For example, we could run high-density SNP profiling. You will again figure that out here in the next slide, including, and we'll run them on Illumina or the Affymetrix platform. We also do a little bit of next generation sequencing. We just started that a few months ago. And then we can also develop custom content for people using either the Illumina or the SequeNome platforms. Again, what that means also you'll know since if you're not in the sciences, in the next few slides, you'll know that as well. So we also do some identity and diagnostics, which are microsatellites and ELISA, qPCR based. The whole idea there was that if somebody was going to send us a sample to do a DNA test, can we get a clinical test? For example, foot and mouth disease is a big thing in the news in the last 2-3 days. So if somebody were to send us a hair follicle, the question is, can we test for DNA for the cow's trait? And can we also look for foot and mouth on the same sample? And the answer is yes. So these are the kind of samples we'll get. Everything from hair follicles on the left. By the way, when you're an entrepreneur, you have to come up with ways to solve problems. So that little device you're seeing on the left is something I designed and patented. So you can send hair samples to us. I thought we'll sell 30,000 of those a year, but we do millions a year, actually. It's incredibly funny. It's a bunch of paper with some plastic on it, but it does the job for us. So samples like cards with blood and semen or nasal swabs, ear tags of an animal, semen straws, notches of ear, blood, DNA, plants, anything we'll take, and we'll get DNA out of it. Those tags that you're seeing there, again, as an entrepreneur, you have to come up with solutions. People would send us ear tags. Those are tags that when you apply on an animal's ear, you can take a biological sample at the same time. So when we got those, it was hard to poke with a screwdriver the tissue out of it. So I designed that little machine down there with a friend of mine in town who's a good design engineer, and we made that little device, and you can see you put tags in a little tray, and you push it into the machine, it'll punch it automatically for you into a 96-well plate. That white thing under there is a 96-well plate. You can do 96 samples in 10 minutes. We have done millions on those machines. We have 2 of them. That's what our lab looks like. Anyway, showing pictures helps you see things better than talk about it. So the left side is the DNA lab where everything is isolated. The veterinary diagnostic lab is on the right. And whatever it is, whatever thing we do, we eventually get DNA, RNA, or protein out of it. Most of them are automated, like the little blue machines that you're seeing. They're all automated DNA isolation machines. Eventually, whatever we do ends up in a 96-well plate, like that's shown there. This is the way we started, again, from a business point of view. When we started with old-fashioned agarose gels that anybody would run in a little lab. We would look at the gel by taking a picture of it, and we'll sit down and score. Why am I saying this? Because those days, in 1998 and up to about 2003, we would sell a data point for about $35 to $40. And we had the means with those little boxes and one automated sequencing machine like the one you're seeing down there. We have now 17 of those. We were able to generate 14 data points also for $40, okay? Then fast forward real quick. Today, that's the DNA isolation lab. It's the biggest group. That's about 30 people working there. Millions of samples are handled by these kids. They put them in the 96-well format as DNA, RNA, protein, and that's get into this lab, which is a veterinary diagnostic lab for looking at diseases, or it'll get into this lab where the DNA is then spotted onto a chip. That blue thing on the very right-hand corner is a silicon wafer. And on those, you can see dots. Those are actually 384 dots on which you can put 384 DNA samples, or you could put 10 dots for one sample. It's up to you. But for each dot, you can get about 50 data points. That's 50 genotypes. And that's run on a mass spec. Those machines that you're seeing, there are 5 of them we have in our lab. They're MALDI-TOF machines. Fancy name for a mass spectroscope, which is matrix-assisted laser desorption ionization time-of-flight mass spectroscopy. It sounded really complicated. All it is that you shine laser on that spot, it ionizes the DNA, flies in a tube, and you're measuring how fast it flew, okay? That's the mass. That's what you're seeing as those little peaks over there. These are the things we do. Because of time, I'm just going to fly through it. We do with those kind of technology. And if we need more data points, from those machines can do about 400 data points economically. And from there to million, we use this platform, which is the Illumina platform. Same idea. You see the little glass slide. Everybody's familiar with that. Those strips that you're seeing there is one sample. We have 80,000 data points you can generate on that little snip per sample, multiplied by 20 redundancies also built into it. So there's a lot. So if you look through a microscope, those dots that you're seeing there is what those strips will be. So you can imagine how small that is. But that's how you generate data. In fact, Sam has the same machine here in Kochi. So we generate our own data from our own chips, and we also buy it off the shelf from Illumina. All the black things are from Illumina. All the reds are the chips that GeneSeq developed, and we sell them as well. So you can see we're now moving into mouse, eucalyptus, custom from shrimp to wild exotic animals like Alaskan bear to deer. Okay, so we have all sorts of chips that people use. And why do they use them? It's all to improve traits in animals so that production increases. In fact, these technologies have completely revolutionized the dairy industry in the world. And now we are focusing on the beef, and it's going to change the entire beef industry as well. Not in India, but definitely in beef-producing countries like the US, Brazil, and Argentina. There's huge implementation going on. So we get samples from around the world sent to us. Just about every country that does this comes to Lincoln, Nebraska, which I proudly always boast about. Okay, so I have another 5 minutes, it looks like. So I want to just talk about, this is what I really wanted to talk about. The rest was just an introduction. I thought otherwise I'll be being rude. But incubating an idea is like incubating an egg. So you select your number one idea or your number one egg and you incubate it. So I want to share with you some of my experiences. And the first message is it's okay to fail. Particularly this is important in Kerala, I think, because people are afraid of failure. It's through failure that you learn. I started 4 companies. I succeeded in one, average on the other one, and 2 I failed. Big deal. You'll hit a home run one day. And now I'm on the next one already. I'm already cooking up plans to start the next company. So you keep on going. So what would you do if you were to start a company? This is my message to you, is that if there's a need, you need to find out that need and ask yourself a question. Can I meet that need, and can I do a good job with it? And is this need within my expertise and comfort zone? In other words, if you're a biochemist, don't start a restaurant. If you're a catering guy and a degree in, you're a chef, you start a restaurant. What do I need to do this, and what do I do to meet the need? Okay? So that's a big question. And if I can meet the need, will it generate cash flow? A lot of biological science people don't think of that. They think of just getting venture capital and starting a company. I'm old-fashioned, the old in Kerala way, let's say, can I generate cash flow? And after you think through it, you write it up. Nowadays, you don't need to write it up in a lot of papers as a big fat business plan. You could do the whole business plan as a PowerPoint presentation. That's most probably that's what a lot of people would like to see. And just because you like an idea, that doesn't mean that the market needs it and likes it. Don't forget that as well. A lot of people think that my idea, oh my God, I'm going to take off. No, the market may laugh at it. Okay, so early stage, you can see the egg is now incubating slowly. It cracked and fell down sort of, but it cracked. So once you take the plunge, you move slowly, avoid risk, learn and grow. Again, from my own experience. This is big time from my own experience. Keep borrowing to a minimum and work with cash flow as much as you can. Also keep costs down. Eliminate unnecessary overheads. For example, until Neogen acquired us, we didn't have a sales or marketing team. So it kept our expenses down 30%. Believe it or not, we found out that's what it is. Focus on what you're good at. Let others focus on what they're good at. In other words, if you know how to manage something, don't be a manager of everything. If you know how to do biochemistry, you do that. Let somebody else do the cleaning. Stay focused on one market. Do not try to do everything. This is a huge problem in India. Everybody wants to do everything. Oh, I can, oh, there's an opportunity there, you run after it. No, stay focused and you'll succeed. Okay, so this is almost done, guys. Once past that stage, if you can see the chicken is now hatched, you can start hatching your own eggs, or if you make a mistake, you could end up being eaten by somebody. So that's the whole message there. Yeah. So build infrastructure and expertise patiently. Too much growth, too much investment, they end up being eaten. Both are dangerous. Follow the 3A principle. This is my 3A principles. Adjust, adopt, and adapt. To what? To changes in the market, changes in technology, customer perception, needs, and demands. Okay, and then don't forget, you will have people who work for you. Don't forget them. Because what a company is nothing but a group of people. And surround yourself with good people, not S-men, who will agree to all the nonsense you have come up with. Advise, correct, criticize, encourage, and support your employees. You nurture, train, develop, delegate, and empower them. That's what I do. I always told my employees, my goal is to sit in a coffee shop, drink coffee, not to run the company. You run the company. Boy, I succeeded. If that's success, I'm very good at it. Create a management team that translates your vision to action. That's also very good and very important. And create a succession plan. A lot of people start companies, there's no succession plan. They don't know what will happen if they die. You got to think through that as well. Okay, well, I'm almost done. As a leader, as a company starter, you have to have a clear vision and you got to communicate that vision to employees and others. And you have to create a workable and fun environment. My employees would cry if they were to work for somebody else because they thought it was better to go to work than go home because it was fun to hang around. You accept advice, correction, criticism, encouragement, and support from employees and advisors. And keep in mind, I said employees. They will criticize. If you're not willing to listen to them, there's no way you're going to make it in the technology business. Make sure everyone knows who pays the bill. That happens to be the customer. Because a lot of time employees think that you pay them. Actually, you don't. The customer pays. Okay? So that's the only reason you exist. You do not drive the market, the customers do. Otherwise, what will happen is if you're not leading like that little geese, somebody's going to shoot you down and that dog is going to hold you as trophy. That's what the whole message is. Thank you very much for your time.
Thank you very much, sir. Now, ladies and gentlemen, our next speaker, Dr. S. Vidya Sagar, Assistant Professor, Department of Radiation Oncology, University of Florida. Dr. Vidya Sagar earned his medical degree from Kerala University, PhD from Manipal University and Christian Medical College, Vellore, and he has completed a postdoctoral fellowship in gastroenterology from Yale University. Dr. Vidyasagar's research include development of a novel method to identify biomarkers in the bloodstream to estimate radiation exposure, the identification of molecules that provide genetically based protection against radiation hazards, and develop nutritional mitigation products using electrophysiological approach to mitigate radiation and chemotherapy-induced gastrointestinal toxicity. He has a National Space Biomedical Research Institute funded grant to develop products to mitigate nausea and vomiting in astronauts during extended space mission. Dr. Vidyasagar's lab focused on understanding the electrolyte transport defect in acute diarrheal diseases and in patients with inflammatory bowel diseases. Now, may I request Dr. S. Vidyasagar to give his presentation. Thank you.
Thank you for the right introduction. And I also want to thank Sam for inviting me here for the presentation. And it is quite different from what. So the talk today is going to be a little different from what you have been hearing. And my experience from bedside, we had actually taken it to the bench, and then we are taking it back to the bedside. So even though the title here says bench to bedside, the story is actually from bed to the bench and then back to the bed. Now, the story that I'm actually going to tell is that a single molecule like glucose, we could actually start off, we had 3 spin-off companies which we actually created. So there was a saga involved in it because we went through a lot of hurdles. We burned our fingers, and there was a time when we had to sell it off when it was becoming really hard. And because my experience was not to start companies, because I'm a scientist, I'm a doctor, but I had very little experience running companies. So the experience that I learned is what I'm going to tell here. So right now we have a company, it's a science-based healthcare company focused on commercializing proven proprietary amino acid-based technology platform to promote healthier living. Having said that, most of the compounds that is there in the product, we wanted to keep it as a low risk. It's considered as generally recognized as safe. It's orally consumed. It's a nutrient product. It promotes clinical outcomes, better clinical outcome. And how we targeted this was that, mostly for patients undergoing radiation and chemotherapy. You know that when patients undergo radiation and chemotherapy, they develop gastrointestinal toxicity. And what I did here, again, is I was using my past experiences in seeing patients who actually have undergone radiation and chemotherapy, and they develop nausea, they develop vomiting and diarrhea. And we wanted a method by which we could actually mitigate it. And the story is going to be from there. So what we created, this product is for cancer patients. We can use it in acute diarrheal diseases. We have small missions in 2-3 countries where we give, on humanitarian grounds, we try to treat diarrheal diseases for kids under the age of 5 years. Then we also target IBD, that's inflammatory bowel diseases. And we have a space program where we are actually producing a drink finally maybe it'll come out as a paste or a gel that is for extended stay or extended mission, where the NASA is actually planning for a Mars mission. They are expecting a trip from 5 to 500 days to a 1,000-day trip. They want a healthy human being at the end, and they want them to come back too. And we also have a product that is now used by the US Army. The story here, so I'm going to tell you a story for 20 years. I've been studying diarrheal diseases. I studied cholera, I've been studying typhoid, and most interestingly, we were trying to look at how can we rehydrate a patient. As you know, in cholera, in the first 24 hours, you will lose more than around 14 liters of fluid, and human body has around 7 liters. So if you don't push in fluids or rehydrate the patient very fast, the patient will die. So the point here is how fast can you rehydrate a patient? So this picture, I'm not going to be very descriptive here. The thing I wanted you to really understand from this was that you should have a strong passion for what you're actually doing. And my passion was diarrhea. So there are a lot of people who actually call me. He's a poop doctor, he treats only poop. He handles poop. So of course I handle poop, and the story begins there again. So here we are looking at the small intestine, and most of the nutrient and electrolyte absorption, everything happens in the small intestine, 80%. And very little of the fluid you can actually see is getting into the colonic segment. So colon plays a very small role in the entire electrolyte absorption, and there is no nutrient absorption in the large intestine. So our job is to see that if you are radiating a patient or you're giving a chemotherapy, if a very small segment of the small intestine is getting affected, it will be a significant volume load into the colonic segment, and you can have diarrhea. And that is what you see in patients. I won't complicate this, and I will try to keep it as simple as possible. So we needed a platform before launching a company, and these were some of the mistakes that we created before. So we wanted a clean platform which will entice investors. So we were looking at what's the kind of knowledge we have. So our knowledge was we can study electrolyte transport, that is how sodium and chloride is getting absorbed in the gut. We could study how nutrient is getting absorbed in the gut, what nutrient absorption can be affected by in a patient who is subjected to radiation. So in this picture, if you look at this, the finger-like projections, those are the areas where you have the nutrient absorption or the sodium and chloride absorption. The deep down, where you put an arrow down there, that is actually the crypt, and that is the chloride secretory region, and that's where the diarrhea happens. So whenever chloride secretion happens, water moves as a result of osmosis, and then you have diarrhea. When you have absorption, sodium and chloride absorption, fluid is absorbed, you have absorption. Now, what happens during radiation chemotherapy is that the fine finger-like projections that you see actually disappears to various degrees. So you don't have an absorptive compartment, and you have only the secretory component. So what we found was that it's not just that absorption is getting defective. What we also found was that there's a calcium-mediated process which produces a chloride secretion. So there's a calcium-activated chloride secretory pathway which we actually found. And now the next thing we wanted to see is that is that something that we can see that as a protein that is coming onto the membrane that we can use as a biomarker? So we started a biomarker company based on that. And this chloride secretory thing, in cystic fibrosis, there's a disease condition seen mostly in Caucasian population. They don't have a chloride secretory pathway. So they don't have a chloride secretory pathway. So what happens is that the mucus in the lung and the trachea is very hard, inspissated, and they cannot cough the sputum out. The mucus doesn't come out. So because they have a chloride secretory defect. But in radiation or with glucose, what we found was that it enhances the chloride secretion. So all we did here was that take that glucose in small quantities, we nebulized it, and we gave it to the patients, and we could see that they could bring out alternate chloride channels on. So this was the birth of our company. So we had an N-trade story. So in the center, you can see what we named as NO1. That was a protein that we were looking at. And that was a protein that was getting activated by glucose. Radiation was activating the same process. So when NO1 is a calcium-activated chloride channel, it activates chloride secretion, and then that's how you have diarrhea. And rotavirus, which is childhood diarrhea, seen in kids less than 5 years of age, is again calcium-mediated, increases anoctamin, produces chloride secretion, produces diarrhea. So in one of the salvage programs that we actually created was that how do we prevent diarrheal diseases in kids? So the simple thing, the rehydration mechanism that is currently adopted or gives a standard of care in most hospitals is that they give a glucose-containing ORS solution, oral rehydration salt solution, that has saved millions of lives. You would have heard this in news and television channels, if, salt and sugar saves millions of lives. Yes, it did. But what people didn't know with that was that this glucose can also secrete chloride. And when you have an absorptive defect, glucose is not a right answer. So rotavirus produces villus damage. The finger-like projections I showed you, it actually damages that. Under those conditions, glucose should not go into those kind of kids. So if that is true, can we rehydrate these kids faster by any other method? Yes, you can, by just withholding glucose. So you can give an ORS, which is glucose-free, and that gave birth to our new drink. Now, again, forming a company, again, it's a learning process. So my learning process was having a clean platform, and how do you maximize the value for your platform? So as I said before, we had a company that we had a product which is of high value and a low clinical risk. Means it didn't want a FDA route. We didn't need to go through an FDA route. We classified them as a medical food, and that was the classification we used. So that you can see, hydration, it could be a $10 billion company. And cystic fibrosis, which we developed as a drug, can be a $5 billion company. And the toximeter, because there is no, the patients who are undergoing radiation chemotherapy, you have no idea when the patient will develop a GI toxicity. Means you give fractionated doses of radiation or chemo to patients. So they come every 4th or 5th day to the doctor's or in the clinic, and they get the radiation, they get the chemotherapy. But we have no idea when they are actually going to develop a GI toxin, when to withhold the drug, or when to say that is it, or when he is actually not going to tolerate the drug anymore. But if you have a marker that will tell you that, or predict that the patient is going to develop GI toxicity, that is something that is needed in the market, and that's a biomarker we had. So we had, in the last, this 2013 was a good year for us. We had 13 patents from our lab. 4 of them actually we were able to commercialize. And to have all this, again, you need a proven team. You need a scientific team. In addition to that, we needed a strong advisory team. So we got some of the best people from this planet. The guy who wrote our textbook of medicine, Henry, he was like our godfather. We grew up learning his subject. When I was studying in CMC Vellore, I was studying gastroenterology, and I thought, I should go to his lab and work for some time. So I wrote to this guy and I said, I want to come to your lab and I want to work. He said, you don't have money, and we don't have money either to come here and work. So write some fellowship, get some money, and if you have money, we'll. So I wrote into Wellcome Trust a career development award. Fortunately, it was awarded. So I went to his lab. I worked for 3 years. From there, we made 2 instruments to study bicarbonate transport. So if you actually look up the profile, you will see that we studied extensively the bicarbonate secretion. And now, 10 or 12 years later, now we have him in our board, and we formed a team. So to get, we have a scientific idea, we have certain things that we think we can take it as a product, but still we need a good management team. And this is something that in my former companies I didn't have. So we got people with experience who can bring a skill set into our company. So we got a CEO who had a exceptionally strong skill set, like 25 years of in a biofuel industry, not anything to do with the medical industry. And again, we are making a drink. So I had no idea how to make a drink and what should go into a drink, what is a supply chain, what is a distribution network. I had no clue on that. So I had to get a guy who had a track record of having done that. So I got the guy, his name was Dan Dennison. He's the guy who introduced Diet Coke to this planet. So I talked to him, I convinced him, you know what, I have a product like this, are you interested in joining? And he said, let's see. So we formed a team. So he brings his Coca-Cola company's expertise into my company. And then myself as Chief Scientific Officer, because I still like to do science, and we have a good science lab where we focus on diarrheal diseases, radiation toxicity, some biomarkers. Then we have Paul O'Knife, he's the director of our cancer center, so we are all part. Then we wanted a team that can go ahead and market it. You know, we have a CEO, we have a man with a vision who could bring a drink to this market, but we still need a marketing team. So we were looking for people who had experience done that. Again, through Dan Dennison, we got the guys who were like former directors and associate directors in Coke who had marketing experience. They had a spin-off company called Bionology. So we got them into our team and we sort of had a proprietary license with them so that they will only work for us for next 3 or 4 years. And that's a deal we stuck with them. So that was our management team. So in the last 2 years, you know, we want to know whether this product will survive in market. So we had to test it out. And we, you know, first we bought this product out as an unflavored product. We sampled out around 25,000 bottles. Some people liked it, some people didn't like it. Some people said, "Why, it tastes nasty, we don't want to take it anymore." But some people said the diarrhea stops. They liked it. So I'll give you one story. So this patient, he actually came from Australia. His wife had pancreatic cancer, head and neck cancer, or the head of the pancreas. Her pancreas is removed. She had torrential diarrhea. She was actually on diapers. And she had 165 chemos. And we were just trying to make this, and it is still not a product. So someone said, you know, there's a guy upstairs, why don't you go talk to him, because he's trying to make something for poop, and he will control the poop for you. So this lady and this gentleman come to me, and they said, you know what, if it is still ready, can we try it? So they tried this. So the diarrhea stopped in less than 24 hours. They became a big fan of ours. And he said, you know what, I want to help you with this. I would like to take this product into Australia, and I'd like to market for you. It is a good sounding business, but again, I don't know business. So I need a good business and a management team to actually tell me if this is the right business proposal. So we started negotiating with this guy. So that is how we started spreading our story, because you have a passion for a product, you are strongly convinced that the product is good. I think then you have a market, and then it likely succeeds. So why did we succeed with our product? Because N-Trade, as we call our drink, it decreases nausea and vomiting. So NASA in NASA, the astronauts, when they go in space, they don't have diarrhea, they have nausea and vomiting. And again, putting nausea and vomiting in the same line is actually wrong. Nausea is completely different from vomiting. You know, the best cure for nausea is actually vomiting. You throw up, your nausea disappears. So, and there are two different events, controlled by two different mechanisms, but you should not say them in the same word. But the thing is, we were able to control nausea. So that was a big success for us. So NASA liked our product. N-Trade, it decreases leaky gut under stress. So this is again a thing, you know, under stress means, you know, people who run, say, marathon, you know, they'll have a feverish feeling. That is because the gut suddenly becomes leaky. You have viral fevers, you know, you would have heard that people, they have lactase intolerance. That's because there is a villus atrophy. The lactase is associated with the lactose, which is needed for the lactose metabolism disappears. And then you cannot tolerate milk. So those things we were able to correct because we were able to tighten the mucosal barrier. So once you tighten the mucosal barrier, anything that is there in the gut lumen cannot go into a systemic compartment. And then endotoxemia will not come. So there's one of the simple way to check whether this product works or not was just measuring the plasma endotoxin level and body temperature. Means the bacteria doesn't go from the colonic segment into the systemic compartment. So in a colonic segment, you have gram-negative bacteria, they produce endotoxins. So that is something that we could easily measure, and we knew that was a good clinical endpoint. And that's what we started using for our clinical trials. Then, in terms of the ultimate point is whether the patient is feeling good. So that is reflected by a weight gain, and and they will have a feeling of well-being, and they survive, and you can use a higher dose of radiation to actually destroy the tumor. So that was something that we could achieve. Starting about cystic fibrosis, as I said in the very beginning, cystic fibrosis, you don't have a chloride secretion. But here we had a simple thing like sugar which could secrete chloride. And we had this rotavirus that produces chloride secretion through a, let me just go right here. So this is it. This is a protein. This protein enhances chloride secretion. So what we did was we combined the Asst.Prof, that is a non-structural protein, an isoform 4, along with glucose to produce chloride secretion. So classically, this is what happens in cystic fibrosis, the current treatment. You can induce chloride secretion, but it is not sustained. They will go rapidly up, it will come down. So we needed something that can sustain a chloride secretion for a period of time. So look at this, what we could do with glucose. So then what we did, if you use glucose and combine with Asst.4, the chloride secretion is sustained. Unfortunately, I think I can't play this. There was a movie I actually wanted to show you that we could enhance the net calcium release inside the cell and activate a chloride channel. So this chloride channel, so this picture that you can see, there's A and B, C, D, and E, and F. So A and B is actually the normal tissue. And you can see that it doesn't come up on the membrane. But C and D, we give 5 gray radiation. Gray is a unit of radiation. So from C and D is 3 gray radiation. It comes onto the surface of the membrane. And that is a key point. So now we go up to 5 gray, you can actually see that the total protein on the cell membrane increases. So, but you cannot take a biopsy from a patient every time you go for chemo or radiation. So you need to have something in the peripheral or in your blood that you can actually use it as a marker. So starting from 0 to 9 gray, you can see there's a dose-dependent increase in the protein expression. And then we were able to see the same protein in RBCs and WBC, and that we made that as a ghost, and that is what we use as an ELISA. So we made a kit out of that, and we gave it to the Department of Defense because they are worried about dirty bomb and things like that. And they tested it, they liked it, so we have an ongoing project with them. So right now, how to take this thing forward, because I am not an engineer, I can't devise an instrument, but I can, we have a chemistry that holds the whole reaction. So we wanted to develop this further. So as phase 1, we thought we'll complete the ELISA, we completed. As phase 2, we wanted it to automate. We wanted this to be at everyone's house who is undergoing radiation or chemotherapy. Every cancer patient, we wanted to have it. So how do we make it? We wanted it to be like a point of care device, something like a glucometer. They can hold it in their hand, put a drop of blood, and read out. They say they're okay, not okay, ready for radiation, not ready for radiation. That is the kind of readout we wanted. So I was looking for a lot of companies that can actually help me with this, and luckily for me, right in Trivandrum, we had this Excel group, and some of the team, I don't know if they're here or not, I actually met them and they said, you know what, we can help you design this. So actually we are negotiating with them for making that as a prototype. A strong IP. So having said, talking about science is great, but if you cannot protect your science with a strong patent, then you are out of business because people will copy in no time what you do. So that's exactly what we did. So we made sure that our IPs are well protected, and we brought in some of the best attorneys in this planet that we could afford and made a very strong patent applications. And we also created strong academic and clinical research links for doing the clinical trials. And we also created a strong advisory board. Now, I was also told by Sam that I should tell, when I'm talking here, what kind of opportunities can Kerala get in similar scenarios for actually undergoing or studying in science or doing biology or doing medicine. So I think by interacting with the people and the students here, the thing that I learned was that there are enough institutions here, like CSIR is there, we have Rajiv Gandhi Institute of Biotechnology, TBGRI, and a lot of other institutions are there. So I think Kerala we can do a lot of things out here. We have an Ayurveda hub, it's 3,000 years of data bank. Every plant is medicinally classified, disease-based classification of plants. I don't think you can get that resource anywhere in this planet. We can utilize that. So if we want a cure, we want a plant, we know which plant we could use, we know the extract, we know the extraction processes involved, and that is something that we have been dedicating in my lab, and we have been successful when we are still working on it. Technopark is here, we should utilize these resources nicely. And then we are internationally well-connected scientific communities right here. The NRIs come here, and and I think they should actively participate, and we should utilize them. There should be one-to-one interaction and see how they can help the kids get what they want. Having said that, but if you don't have an innovation platform and an innovation hub, things are not going to work. And I think for a dedicated biotechnology, we should still have an innovation hub. And I'm not going to go through the list, but one of the two core things that I found is lacking in this country is that we don't have an animal facility. The number one thing that should be there in this state for a good biotechnology plan to come up is a good animal facility. No animal facility, no research. Biostatistics core, we should have a biostatistics core, and rest this, I think everything is common. I won't go into this either, they're all standard. Questions?
We'll do the questions, and yeah, thanks, Dr.
Thank you very much, Dr. S. Vidya Sagar. Now, ladies and gentlemen, our next speaker, Dr. Ramesh Hariharan, the founder and CTO, Strand Life Sciences. And he's the adjunct professor at the Computer Science Department at Indian Institute of Science. Ramesh's research contributions include the fastest known algorithms for several fundamental algorithmic problems, including graph classification, tree pattern matching, approximate text searching, etc. As part of the Strand, Ramesh has been leading team-building analytical tools for high-throughput molecular profiling. Ramesh got his bachelor degree from IIT Delhi and his PhD from Current Institute of Mathematical Sciences, New York. Subsequently, he did a postdoctorate at Max Planck Institute, Germany, then returned to India to take up a faculty position at Indian Institute of Science, Bangalore. Ramesh was also one of the founders of the Computer project aimed at creating a low-cost handheld device to bridge the digital divide. Now may I request Dr. Ramesh Hariharan to give his presentation. Thank you.
Thank you for that introduction, and thank you for inviting me here. It's a pleasure to be back here in my home state. I want to talk about a theme that is very common in the US, but it's been traditionally missing in India, and that's the theme of bridging academia and industry. So Strand Life Sciences was founded in 2000, about 13 years ago, as a spin-off of this. What you see, the photograph there, is the Indian Institute of Science campus, a very nice, verdant campus in Bangalore. The motivation, and all of us founders were at the Indian Institute of Science for a few years before this, and the motivation as we started observing things around us was that, there's a big chasm between industry and academia. Academia, the institutes of technology and science and all of our universities, they work on deep problems. They know how to solve hard problems. But in the process of trying to solve hard problems, they become very narrow and often out of touch with the reality around them, the applicable reality around them. Industry, on the other hand, tends to be very short-term focused, trying to solve what is of immediate necessity, and often lacks the ability to solve hard problems. And there's a whole opportunity in between that requires solving hard problems and yet addressing relative near-term goals. And that was completely missing in India. I think it continues to be missing, even though there's been substantial progress happening. So our goal was to see how can we bridge that chasm? How can we create an organization that is rooted in academia, but is driven by the forces of business, the forces of industry? And that's what we have succeeded is still too early to say. We've certainly done well for the last 12 years. We've contributed quite a bit, and I'll show you what we've contributed to the world, but our eventual success is still to come. What did we want to do in this company? We had, just as we wanted to bridge academia and industry, there was another goal that we had, is to bridge two very different disciplines of learning. The traditional biology discipline and the traditional computer science discipline. Most people who go through high school pretty much very quickly decide that they don't like biology, or they like biology or they like maths, and then the split happens. And then that's a split forever in your life. You never come back and look at the other side again. There were lots of developments happening in the world which essentially required that people had to come back and look at the other side, that these two disciplines had to come back and meet each other and work together. I myself took the computer science route, the maths route, and decided never to look at biology in my 11th class, only to, by the time I came into my PhD, biology certainly started looking very attractive. And the reason why it started looking very interesting was traditional biology, the way we were taught, is all about now how does a frog look? Can we cut it open a little bit and see how the organs look? And what are the millions of different species, and write 10 properties of each of those species? That's a very outdated view of biology. The fundamental view of biology from the inside out is how does life work? How do we work? How do our bodies work? How do our cells work? And that's at some deep internal level driven by a molecular system, which is a computer. It is a computer that has its own language of description, that has its own way of executing jobs and tasks. And if you start looking at it as a computer, you suddenly start getting a whole new different set of insights as a computer scientist. And you suddenly start seeing that these two worlds are not that different. The biology world is, though, infinitely more complicated than the computer science world. So what we do at Strand Life Sciences, to get back to the point, is to look at as the world is evolving, newer and newer methods to look deep into the computer that each cell runs. And that computer is made up of many, many different molecules. There are hundreds of thousands of different molecules, so-called genes and proteins and so on. And as the world is getting better and better at looking at these molecules and trying to understand how these molecules work, it also generates a lot of data. And that data requires a lot of crunching before you can get down to an answer on how the biology works. For instance, if you walk out on the street and inhale some smoke, then and there's a lot of smoke on our streets out here, your lungs will react. Each cell in your lung will react in a certain way. There are 20, 30,000 genes in your body. Each of them will react in a certain way. Some genes will kick up their activity. Some genes will kick down their activity. As a result of consistently kicking up or down that activity over a long period, let's say, the whole state of your system will get moved from one state to another, and that could eventually land up in a disease state, etc. How does that happen? Which genes go up? Which genes go down? All of that is something that now can be done very easily by merging these two disciplines and running appropriate experiments and crunching that data and getting insights. To do all of that, you need a team. And as you can see, we are trying to bridge academia and industry. We are trying to bridge two different disciplines. And so you need to build a team that has strong capabilities in both disciplines. And so you need a team of computer scientists and a team of life scientists. And as I told you, the divide happens very early in life. By the time you're 11th or 10th, you've already made up your mind to go one way or the other. And now you've lost the ability to look at the other side. And now you have to bring that back. And you have to bring that back. There's no academic institutions in India that do a good job of bringing that back. All that effort is just about in its infancy and starting. So we have to do it in an industrial setting. So we hire people and do that. So which means we have a pretty large team now, 200 people, 40 PhDs, 65 people from computer scientists from the IITs, about 30 odd PhDs, molecular biology PhDs from various universities. So it's been a very interesting exercise trying to make them all talk to each other, very different lingos, very different behaviors and expectations, and to get them all talk to each other and work together towards building real products. And that's something that we would certainly count as one of our successes. And what has all of this translated into in terms of our impact? The impact that we've been able to make on science is small, but not insubstantial. So all the tools that we've built to allow people to look at, molecularly inside a cell and understand what is happening in the biology, what is happening when a cell becomes cancerous and how does the cancer develop, how do certain genetic diseases happen. The tools that we've built to understand all of this have been used in more than 10,000 publications across the globe. Scientists who run their experiments use our tools and then publish using these tools. If you search on Google Scholar, you'll find 10 to 15,000 publications which talk about all the tools that we've, all the papers that have used our tools to generate fundamental new insights into science. So that's a good contribution, but that's a limited contribution. We have to reach beyond scientists. We have to reach society, and that's how do we reach society? And that's been our effort over the last year or so, all of this scientific knowledge and the tools that we are building to delve into the processes of life, can they be used in a clinical setting, in a medical setting? You saw several examples here where in agricultural or sort of veterinary settings and in medical settings. And here is yet another example of how deep understanding of the genes and of the molecular basis of biology can be used in a real example to help real patients. So here is a family, and this is a case from around Bangalore that we've one of several cases that we worked on. A family where there were two children. They were born with all kinds of problems, hypertension, which you don't expect in a newborn child. It typically develops as you grow older. But in addition, they both died in the second year of life due to respiratory insufficiency. The natural question that the parents would have in such a situation is, the parents are still young, they can have the next child. How do I know that the next child will put all our effort into it and then spend a year and then the child passes away and that's a tragic outcome? How do we prevent that outcome? Do we have any tools in medical science that will enable us to prevent this outcome? Medical science, is still a very heuristic science. It has a limited set of tools to be able to solve problems like this. But the good news is that the tools to solve problems like this have been evolving, and now they're reaching a level of maturity. So we were able to actually take this family, go into their DNA, sequence the entire DNA. When you sequence the DNA, each person's DNA is different from everybody else's DNA. So your DNA and my DNA will be different at millions of places. So now this problem that these children have has been caused by one particular point in the DNA that they have a mutation which causes this problem, which causes them to die in year 2. So they have a mutation, a place in the DNA. A DNA is 3 billion long, and of which there's one place where they're different. And we have to get to that one place somehow, and then figure out how we can help the parents next time around when they have a child. So the interesting thing is, even though this looks like a formidable problem, the tools to read the DNA have pretty much matured, and that's causing a sea change and causing a lot of entrepreneurship opportunities that Sam is working on, that we are working on. So the tools to read the DNA have matured. But that leads to a whole lot of data. The tools to crunch that data, the tools to bring all the medical and the clinical and the biological knowledge to bear on that data to be able to take millions of these differences that these children have with everybody else and then narrow down to that one difference that is causative in this case is something that we've been building quite a lot over the last few years, and now that's also reached maturity. So we managed to identify that one variant that is causing one mutation that's causing a problem in these children. And then when the next pregnancy came about, we tested the fetus, and luckily the fetus did not have that mutation, and so we counseled and said, this fetus looks like she should thrive. And the girl was born 2 months ago, and she's thriving, she's doing well, none of these manifestations. So you can look at the power of this technology, the power of being able to look into the biology, take all the data that comes out, crunch it, bring all of the medical and the clinical information published in literature to bear on that decision, and make all of this part of standard clinical practice. So that's what we're trying to enable here. The Holy Grail, of course, one of the big problems that we will all be confronted with, there's going to be, as lifespans increase and as environments become increasingly more polluted, there's going to be increasing frequencies of cancer. Cancer is a genetic disease in the sense that we acquire mutations throughout our lifetime, and when those mutations reach a certain number and a certain level of potency, the cells start dividing uncontrollably. Cancer is not a single disease. Depending on what mutations get acquired over our lifetime, the cells will start behaving in slightly different ways. And so it's actually a collection of diseases. It's a large collection of diseases. It's not a single disease at all. And depending on what mutations have been acquired, the treatments may be slightly different, the prognosis may be slightly different, what you need to do may be slightly different. For instance, I'll give you a personal story. I had an uncle of mine who got bladder cancer. And the standard treatment for bladder cancer is very conservative. You treat with BCG, which is the same thing that you treat tuberculosis with, for instance. So, and most bladder cancer cases sort of are relatively mild, they resolve, but not all. And now medical science, medical practice, until a year or so ago, did not have the tools to identify whether this particular person has a cancer that's unduly aggressive, or is this one of those standard cancers that will get treated by BCG? There was no way for a oncologist to figure out the difference. And so they just decided to treat the conventional way. In 3 months, the cancer metastasized, moved to all parts of the body, and the person died in 6 months. So the recognition that cancers are different, some cancers are a lot more aggressive than other cancers, some cancers need a different sort of treatment than other cancers, has been sinking in over the last 10 years. But the tools to be able to identify these different cancers from each other in medical practice, to be able to implement it, are just about starting over the last year or two. And at Strand, what we are doing is to build these tools, and we've been working with a number of hospitals doing a number of studies where we take these cancers, look into the genetics, and make recommendations of how the therapy can be tailored. In a number of cases, oncologists manage to tweak their therapies and change things around to have a better chance at the cancer. This is still emerging. Over the next several years, these methodologies will refine to the point that they'll become part of standard cancer care, and our ability to fight cancer will improve. But there's a lot more that needs to be done, and this leads me to the last section about what are opportunities for entrepreneurs, what does society need to do in this area to get better. To fight cancer, we need a highly multidimensional approach, and one of those approaches is, first is the recognition that fighting cancer is largely the number of tools that clinicians have is very limited. So they are hitting in the dark a lot of the time. Some of those tools will come about through long periods of research. The other tools have to come from just careful monitoring, data gathering, and data analysis. Transparency into the medical system is very poor. We have to be able to track every patient, every cancer patient, note down what the nature and stage of tumor is, what treatment was given, how they progressed, be able to create these databases on large scale across the country, be able to correlate the certain treatments with certain types of cancer, certain diseases, and as this ability to look at the mutations in the cancer comes about, integrate that into this system as well, and then see if there are trends emerging that will allow clinicians to be able to make better decisions on how to treat cancers, I think. So that's, I think, a big area that I would definitely urge all people who are looking at the medical space, entrepreneurs who are looking at the medical space, to think hard about. With that, I'll stop, and thank you.
Nausea, how do you control them for cancer patients down to the astronauts? And then we heard the fascinating story of Strand Genomics, where they build tools which would help biologists and scientists manage data, and how they are moving from that to create solutions which will help all of us. So we have a few minutes left. So any questions to any of the scientists, entrepreneurs are welcome.
You said about the lack of transparency as far as the medical science is concerned, practicing medical sciences. And you also said about analyzing data to arrive at many of those diagnostics or many of those findings. So how do you plan to bridge this? Is it something that your laboratory can alone do it, or is it something that the authority, the stakeholders, it's such a large call, isn't it?
I don't know how to tackle, and that's why I put it out. It's problems that we collectively need to tackle, and maybe society, government, or independent entrepreneurs need to tackle. We are working on, companies need to work on a relatively narrow slice of individual companies need to work on a relatively narrow slice of focused activity. But society, if it has to solve this cancer problem, it has to come at it at many, many different angles. Pharmaceutical companies creating drugs is one thing, people understanding the mutational profiles, but looking at the data, careful gathering of data, archiving it, and making it accessible to patients and researchers is going to be clearly a burning need. Who can do it? That's a big question. I don't have good answers. I think it'll have to be some intelligent entrepreneurs coming up, some hospitals wanting to do more than what they're doing, and the government together mandating a few things together, I think, making it happen.
Yeah. Since you are dealing with cancer, a particular question. Of late, there has been an increase in the number of cancer patients. Can you pinpoint some of the reasons why this is happening? Almost every second person who we come across died of cancer. What has been the cause for the increase in cancer, and what preventive measures or what steps can be taken by all and sundry to reduce the incidence of cancer?
It's a great question. There are a few points that I would like to say for this. Number one, back in 40s, the life expectancy would have been around 45, 50. You fast forward and you come back to 2012, 2013, life expectancy is now 75, 80. So now you have a population that is growing older and older. And one of the things by which we are sent off from this planet is actually through cancers. So there'll be the more geriatric population that you are going to face, the more geriatric problems, that is cancers, you will actually see. But how do you find and that's an early detection program. And that is something that our state should actually be taking an initiative on, that is colonoscopy. After 50 years, once in every 5 years, do a colonoscopy. You see a polyp, do a polypectomy. And then every other year, do a colonoscopy. You can early detection, early cure. Breast cancer, mammograms. It is mandatory, you should do it. If you have a family history of colonic cancer or you have a breast cancer, you should actually do a mammogram. There are populations where they actually do bilateral mastectomy, because they think they are at risk.
BRCA testing.
Exactly. So there are a lot of tests available. So that's where the genomics are actually coming up. Because once you do the profile and these are the genetic markers that is there, and there's a genetic predisposition, you should go in for screening.
Yeah. Abe would like to add to that.
So since you asked what's the change, like he said, 40 some years ago, life expectancy was about 45 years, something like that. But today it's in the 70s or 80s, right? But things have changed in that time too. The environment has changed. The number of industries that have come up that are new have changed. So the pollutions have changed. So I'm not trying to get into an environmental debate here whether industry is good or bad or we're polluting or green or anything like that. That's not what I'm talking about. But those changes are definitely contributing to the incidence of cancer. At least I firmly believe that. Think of it. You can't eat fish from the sea. The sea is a huge place, but there's so much mercury in the sea that even an average fish you catch, you can detect mercury in it. I mean, that's what you're eating. So you're going to get sick. So the chances of getting diseases like cancer is going to increase as industries progress until we come up with ways to control that and reduce what you're seeing in the environment to what it was maybe 100 years ago.
My contradictory question is, fine, if all cancers growth were, say, 40 plus, but then we find there are also cancers growth from, say, age of 1 or even less than that.
Very true.
And many people who are dying at the age of 30, 40 of cancer.
Pediatric cancer has been increasing. Anything coming up that babies are born?
Mutations, if the mutations are there, and like we were talking about the environmental factors. Talking about environmental factors, I just wanted to add, that's a great point that you added. That's something that I didn't say, but something more to add to that is actually the diet. Historically, what we have been habituated to and what we are doing now is totally different. Give you a simple connection here. Traditionally, we are in the south, we are rice eaters, and in the north, we are actually wheat eaters. They go with potatoes, and here we eat rice. Now, and we mix it with yogurt. And the yogurt actually is a form of a probiotic. Now, the kind of rice that we eat in the south, we have this round plump rice, which is more pectin-based rice. Whereas in the north, they actually take a long grain rice, which is more amylose-based rice. That is more what is called as high calorific, means it is easily digested. So that is not a great rice to eat. So traditionally, we have been eating the round plum rice with yogurt. So now the point is, why do you need this kind of pectin-based rice? The reason we need that is you need a lot of this carbohydrate undigested. It should go into the colonic segment. In the colonic segment, they are acted upon by the bacteria that is there. They ferment it, and they produce substances called short-chain fatty acids. So the short-chain fatty acids are classified as short-chain, medium-chain, oligo-chain, and long-chain fatty acids. The short-chain fatty acids, that is like formate, acetate, propionate, butyrate, they are the nutrients for the colonic mucosa. So if you don't eat good carbohydrate, you don't have the nutrient for your colonic mucosa. So the people who eat high-protein diet, meat eaters, they produce isoforms of these butyrates, and this is more carcinogenic. So the cancer incidence can go up. But that this doesn't answer to your question, why does childhood cancers come? But I think just the mutations are actually increasing because of the way we have been treating the environment. Because again, coming back to the environment, something that we should all remember about is that we talk about plastics, and there is a lot of regulations going on controlling plastics that we are throwing out. But I think the bigger problem that is that we are all going to sit on a pile of crap that is fecal matter. The reason being, it is true. The reason being, we put a lot of detergents, Lysols, disinfectants into the toilet. The thing is, the bacteria are needed, the environmental bacteria are needed to break down these fecal matters. And if you put all this Lysol and things like that into the, and flush it down the toilet, these bacteria are not going to degrade this fecal matter. So over a period of time, our 5-6 generation down the line, they will all be sitting on a pile of
Since you're the poop doctor, it's a great statement.
Talking about the isolation of Ayurveda plastic material you were mentioning, that's a major, it's not a question, it's my observation or my finding. Most of the plants have a tendency to absorb heavy metals from the soil.
Right.
It's causing a major worry to the Ayurveda industry as it is.
That is an excellent point. That is why I was a very serious advocate of actually extracting it, isolating it, and getting the active ingredient, rather than giving it as a pure crude extract.
Yes.
And if you just go back to the NC, that is the National Cancer Institute, back in the United States, they have what is called, this is an era where in silico modeling has come. And they have a stockpile of used or which has been tested in other conditions. They have said that it doesn't have a use. Those compounds have been set in as a library, and it is freely available for anybody to use. But if you look at how many drugs came out of this, not many. One of the biggest, richest resources that we have in this state is that we have a medicinally classified plants. If we want to get answers to most of the disease conditions, we have the answers right here. We should have a platform to go ahead and extract. We should have a processes to actually elucidate, and then you should have animal models, cell models that you can actually use it for screening.
Agronomical practice is also important. What happens, these plants are grown next to the railway line and it absorbs a lot of mercury and zinc. These are the problems, it's a major problem for the Ayurveda industry.
Yeah.
We have a drug industry, which is a big mafia. We have the healthcare industry, which is another big mafia. Will genomics become the third mafia?
Hope so.
As long as they make me the godfather, I am okay for it. Any other questions? Yeah.
Not really a question, just an observation. I really like Dr. Hariharan's take on the divide between biology and mathematics. I'm a class 12 student right now, so I can really feel that. The mathematics I learn in school seems completely devoid of or completely unconnected with what I use in daily life and with biology as such, and I cannot find the link between the two.
I want to answer this question a little bit, and then I'll give it to you. I want to go back into this because, the lab where I actually work, the lab that I actually set up, I use physics-based devices, and I study electrophysiology. But you can see I have a surgery background. I was a surgeon for so many years. So maths and science are related. Now, application, you learn math. And we were talking about with some kids yesterday, and one of the things that came was, you all learn pi. And if you ask them, you will say it's a 3.1 for blah, 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 they will go with an answer. But the thing is, if I give you a piece of thread, can you define pi? Not many kids, actually, they know it is actually circumference divided by. He's good at math. Yeah. He's good at math, so that's a great thing. So, but again, if you talk about a slope, y is equal to mx plus c, you ask about m, what is the relationship m with c, and not many kids will actually say that. So again, what I'm trying to tell here is that a practical application of the math is critical. So for me, my passion was physics, even though I took a medical background. And because back in Kerala, parents tell what to do, and I come from a defense background. My father said, "Do this," I do that, and that was it. We don't talk back, because if you talk back, they'll kick you out of the house. Then there is no money. So that's how things work in India, and even in Kerala. And I think everybody wants either an engineer or a doctor, nothing else. But doctors are the least earning members in the society. I don't know if people know that. They make no money. If you really are,
we can discuss that.
Yeah. So the point.
Doctors' problems.
These are doctors' problems. So what I'm trying to tell here is that, yeah. So your application. So connecting physiology, connecting biology, and with math, they're all interconnected. And you can do a better thing. And that is what I have actually fast-forwarding many years now. All that I learned in biology, I'm actually reciprocating in relation with physics. And I think some of the success that I'm actually getting is because I knew physics a little bit, not that well, but I also knew biology a little bit, and that's the connection I'm having. Maybe you'll have something more to say.
See, I think that thing needs to be very clear to students as well. We need to make it very clear to them, like, I didn't know this. Yeah. And I would be continuing with this mindset that these are two different things. And luckily, on being here, I know this thing. I can tell to my friends as well, I can spread the word, but then it needs to be in a school curriculum as such.
Fundamental change in the way schools operate. Eventually, society has to solve a lot of problems. And to solve those problems, you'll need a bunch of skills. And reading is a skill, writing is a skill, maths is a skill, knowledge about how the world operates is a skill at a physical level, at the biological level. So all of those are skills. Now, the more skills you can pick up, the more you would have in your armory to solve problems. If you have paintbrushes of 20 different types, you can create a much more nuanced painting than if all you have is a single paintbrush. So the more paintbrushes you can pick up, the more you would have in your arsenal to solve problems. And that's the way you have to look at it. There are no sharp divides. Just because you did math for the first 4 years of your college life doesn't mean anything later. It just means that you've learned that a bit more than everything else. In the next 4 years, you might have to learn other stuff. It's a completely fluid world. You move around, depending on what problems you are interested in solving.
Everything is multidisciplinary. Actually, if you listen to the 3 talks here, you'll find out that so many disciplines were all integrated into one. And then just from my own personal experience, I started with the plant sciences in India, went to America, did a degree in molecular genetics, worked in a DNA sequencing machine manufacturer. So I interacted with engineers, software developers. And I started thinking like an engineer and software developer. My company was all animal genomics. Now I'm thinking of starting a medical device company. You have to be crazy. Why? Because you get to know a lot of disciplines and you meet the right people in these disciplines and you get excited about everything. So everything is multidisciplinary is the take-home message there.
Okay, one last question. Yeah, go ahead.
I was wondering that, when you screen only genomes for diseases, like how Professor Hariharan said, you can screen for cancer. How can you be sure that other factors, environmental or individual metabolic system, how do you know that those are not going to affect the outcome? You can't surely say that the disease will happen in a certain person.
That's a very big and loaded question, and it's a question about how, there are many questions in there. One question is, how much does the genome tell you about what is going to happen? Right? That's one question. And if you look at it, the genome tells you only a little bit about what is going to happen. But within that little bit, there are a few bits that are overwhelming bits. There are a few bits that will just, completely overshadow everything else. Those bits you can say with almost complete certainty that no matter what else you do, nothing can change.
Typically called Mendelian diseases.
So
I think that just add a point to this. Lung cancer. There are most of the lung cancer that you would hear is that it is actually related to smoking. So there is a gene that is responsible for that. That means you're already hardwired for that. But if you don't have the gene, even if you smoke, you don't get, but it is in a very rare set of population. So those guys are actually bad examples. They'll say, you know what, I've been smoking for 80 years, I didn't get a cancer. Now why do you tell me that I should not smoke? But that is not correct. Just because you didn't have that particular mutation, that means he didn't have a genetic mutation. So he's not going to get cancer. But if you have that mutation, then you're already wired in. All you need is a switch to put it on. Environmental factors are the switches. So those are like the triggering the metabolites.
Some of the diseases have many genes in the blood, like diabetes or cardio. So their influences are huge. Environment, food you eat, your stress level. So it's a very complicated thing that will keep us all busy for the next 20, 30 years.
Do you think it is possible for us to have DNA identity chips? Like if I have the content of my genome with me, that if I go to a hospital and they could give me medication accordingly, so they know that certain drugs wouldn't work with me and some would. Do you think that is possible?
It will be. No, all of that is possible. All of that is possible. All of that is what some of my cancer allusions were to that if you know what the genome of the tumor is, then you can tailor treatment accordingly. All of that is very, in the last few years, it's been all forming. I think it will take time to mature. Yeah.
Yeah, and it's personalized medicine. Yeah, this is what is actually called as a personalized medicine, and that is actually what you're going to see in the next century.
As soon as a kid is born, he'll be sequenced.
Yeah, but the one thing to warn, I mean, this is an emerging area, so it's not as if there's going to be ready-made solutions that if you get some disease that you go look up your DNA. I mean, that's not. We've got to do a lot of work to figure out which bits of the DNA translate to these sorts of clear treatments, which bits are sort of completely nebulous and that we cannot do anything about. And so it's going to be an ocean that we have to go through. It's going to take a while. But there are examples. There are a number of examples that can be given today. But those are still, I would say, handfuls of examples, not truckloads of examples. We have to generate those truckloads over the next several years.
Yeah. Okay. Thank you, everybody.
No, we need to give
So, before winding up, may I request Sam Santhosh sir to please give away mementos to our speakers, firstly to Dr. Abe Oommen, Director, International Business Expansion, Neogen Corporation. Thank you very much, sir. And now kindly hand over a memento to Dr. S. Vidya Sagar, Assistant Professor, Department of Radiation Oncology, University of Florida. And finally to Dr. Ramesh Hariharan, Founder and CTO, Strand Life Sciences. Thank you very much, all of our speakers.